My pleasure to introduce our lunch speaker. As we mentioned, after a morning full of law, we're going to do something a little different. We're going to do a little bit of science. And our lunch and keynote speaker is Professor Christina Smolke from the Bioengineering department here at Stanford. She is chemical engineer by training. She's doing some very interesting and really remarkable things with RNA molecules and information processing. And she has, despite her relatively junior tenure as a faculty member, won pretty much every award I can come up with. She's won the NIH Director's Pioneer Award, the World Technology Award in Biotechnology from the World Technology Network. She's a fellow of the Alfred Sloan Foundation. She won the NSF's Career Award, and she was named one of the top 35 young innovators of the world by Technology Review. I'm very much looking forward to hearing what she has to say. Christina.
So today I'm going to talk about some work that we've been doing in my laboratory and technology that we've been developing that can ultimately fundamentally change the way that we source and discover our medicines. So I want to start off by reminding us that we have a long history of using biology to make things. In particular, there's a long history of using organisms like yeast in our food and beverage industry, so to ferment, basically brew beer, ferment wine, and make bread. And the products of these fermentation reactions are fairly simple molecules. They're carbon dioxide and ethanol that we're using for those purposes. Now, more recently, let's say over the past few decades, there's been an interest in the research field and also industry to leverage the capacities of biology to make molecules, but to direct the capacities of biology in sort of a synthetic way, to engineer organisms to make molecules that they might not normally make. And I'm showing you here some examples of different types of molecules that have gained a lot of interest in industry, and the companies have been started to make these types of molecules through biological processes, starting at the top with fuel-based molecules or molecules that can be used as fuels, moving toward starting materials in the middle, so molecules that can be used as starting materials to make other types of commodity products. And then finally, more recently, we've seen a movement into compound molecules that can enter into the fragrance and flavor industry. So even if you're not a chemist, what I would just want to sort of highlight on this slide is the idea, hopefully, that as we move from top to bottom, we're sort of moving towards progressively more complex molecules. Meaning we start with sort of fairly simple kind of linear hydrocarbon chains. We maybe extend those, and then ultimately we're beginning to introduce things like ring structures. And so that is sort of how we would sort of categorize maybe complexity. But even the molecules that you see here are fairly simple in regard to the overall kind of molecular space that nature can access. And in particular, what I'm going to focus on today are molecules that would be sort of on the bottom, even lower on this slide, which are molecules that are useful as therapeutics and drugs. And so the next question to sort of think about is, where do our medicines come from? And in fact, over half of our medicines come from natural sources or are inspired and derived from molecules that we find in nature. Showing you here on this slide just some charismatic examples of different types of drugs that we get or have gotten previously from nature, and in particular from the plant kingdom. And what you can see is that there is actually a broad diversity of different types of therapeutic activities that the plant kingdom can make, in particular compounds to treat cancer, viral diseases, to treat heart disease, and even some of our most potent analgesics or pain medicines. Now, over half of the compounds that we do get from nature come from the plant kingdom, and so the plant kingdom is really a very active source of our drugs. And so you can then imagine, though, that when we rely on nature to source our medicines, there are a number of limitations that essentially arise. And I just want to review them with you here. The first is that when you're growing plants as a manufacturing platform, you're typically growing them out in the open environment, and they're growing over long periods of time, typically over the course of a year. So they're exposed to changes in climate, they're exposed to disease and pest, and all of that kind of basically affect the amount and the quality of medicine that you get from the plant any growing season, and that can introduce a lot of instability and fluctuations in the market. The second is that these plants are not making these molecules as medicines for humans. They're making them for their own purposes, oftentimes in defense. And so what that essentially means is that, number one, there's going to be a large molecular space that we're just not going to be able to access from nature because it doesn't have a natural purpose for making those types of compounds. And number two, the molecules that we do get from nature are oftentimes going to be imperfect because the plant is not making them as medicines for human. There's going to be negative side effects or efficacy issues that we have to deal with. And then finally, it's the case, right, that most people currently still to this day don't have sufficient access to the medicines that they need. There are a number of reasons for this, but oftentimes one of the major reasons can be because of the supply chains, because of access to these supply chains and the cost of medicines. All right, so I'm going to focus today on one particular example, highlighting some of the challenges that come up, and then the technology that we've developed to address some of these challenges. And in particular, I want to zoom in on the manufacturing supply chain for our pain medicines. And so what you're seeing here is a field of opium poppies from which all medicinal opioids are currently derived. This is a picture taken from a field in Australia. So this is what it would look like during a certain time of the year where the plants are blooming. And there are just acres and acres and acres of these types of poppy fields. So the first thing you can notice is that even though this is a controlled crop, a drug crop and a controlled substance, we're growing these out in the open, and the type of containment that we have is essentially a small fence with a sign that says keep out. And that's kind of the best that you can do when you're growing this amount of land to produce your crops. So let's talk about the scale that's required to make our medicines each year. There's about 800 tons of opiates that are sourced from the opium poppy to meet the global demand for these medicines currently. What that equates to in terms of land usage is about 100,000 hectares. This is for the legal market. And you can think of this as about the size of Rhode Island. And because poppies require very specific climates to grow, and because they're also very tightly regulated, they're only grown in a very small number of regions across the world, with over half of the poppy crop being grown in Australia today. And then finally, the World Health Organization estimates that about 6 billion people have insufficient access to pain medicines to treat moderate to severe pain. So this is certainly a case where many people in our world are suffering through pain through different types of chronic and late-stage diseases. Now let's look at what the poppy is actually doing. So we zoom in. This is a picture of the poppy plant where most of the compound is accumulating in the pod that you see here with the petals off. And the poppy has evolved to basically primarily make morphine, okay? And so it's making it as a small percentage of its overall biomass. So what you have to do is annually, one time a year, you harvest the crop, then you have to extract the very small amount of this compound from the plant material. This requires fairly toxic chemicals to do that. And then because most of the farming and agricultural farming is being done in areas outside of where the medicine is being used. So for example, the US uses about 70 to 75% of the global medicinal opioids. You have to ship or transport very large amounts of raw narcotic material from countries like Australia into the US, and that adds a lot of cost. And then finally, because morphine is actually not a very valuable medicinal opioid, you have to take the morphine as a starting material, and most of it gets converted to the higher value medicinal opioids that you see here, and the antagonists, like the now compounds that are used to treat addiction. Now, another challenge is that this is a very interesting medicinal plant. It's not just making analgesics like the opioids. It's making a number of other compounds that have value to treat other types of diseases. So for example, it makes a particular compound that is currently used as an antitussive, but also been shown to have anti-cancer activity and activity in treating neurodegenerative diseases, and also vasodilators. And so the challenge here is that the plant is making all of these compounds together in one mixture. And so you have an additional challenge of if you actually want to get access to these other compounds, which are produced at even lower levels, you have to go through very costly and inefficient extraction processes to purify these compounds away from the opioids, in particular because the opioids are controlled substances and the other ones aren't. that really limits the access to these other types of medicines the plant might be making. And then finally, as most of us are very familiar with in this country, this agricultural supply chain does support an illicit market that is thriving either through farming of drug crops to enter into the illegal heroin market or just abuse of medicinal opioids. So we wanted to ask the question of whether we could shift from a manufacturing platform that relied on nature and had these inherent challenges to one that instead relied on biotechnology. And could we engineer a very simple microorganism like baker's yeast to grow on sugar and be able to produce these types of complex medicinal compounds, not within a matter of a year or an annual season, but within a matter of several days? And in doing that, would we be able to address a lot of these challenges? So I want to give you a high-level picture of what we're ultimately talking about here in terms of the vision. We're talking about shifting from the current process, which is relying on one plant, the opium poppy, to give us one compound, and instead saying, we're not just going to limit ourselves to one organism and what that organism happens to make in nature. We're going to go into the broader natural world and leverage the broad biodiversity that's available throughout the natural world. We can do this because of the advances in DNA sequencing and synthesis. So essentially, researchers can go into the natural world and begin to sequence the genomes of potentially any organism. Those genomes then encode the information by which that organism makes certain compounds or the activities that are required to make those compounds. We can basically begin to then harvest those activities from different sets of organisms, synthesize that DNA, okay? And then we can combine those activities from different organisms into a single engineered host, like our yeast host. So now we have an organism that is combining activities that don't occur normally together in nature. And what that allows us to do then is when you combine activities that wouldn't occur normally in nature, you're able to go beyond what nature gives you. You're able to make compounds that you wouldn't necessarily find in nature, but could have improved therapeutic properties or reduced side effects. And now what you have is a scalable production platform where you can take those yeast, grow them quite rapidly on very inexpensive media, basically sugar, and within the matter of days, you can have tons of your product, right? So a very rapid response in production capacity. Okay, so one of the reasons that this approach is so powerful is that the overall vision that I just showed you is one in which we're not limited to just one particular medicinal compound, let's say medicinal opioids to treat analgesics. We can leverage the architecture that's present within nature for these pathways, where essentially you can create platform strains that will produce advanced intermediate scaffolds like you see here. And then from this advanced structure that you see here, you can access all of the types of molecules that you see around this yeast strain. And so again, what that means is you now have a very efficient way, not just to, let's say, address some of the challenges with our pain medicines, but also to address some of our challenges with our cancer medicines, with our antiviral medicines, and et cetera, right? And so this is really sort of, you want to think about it as a broader platform technology that gives us that type of efficiency. Okay, so I'm going to tell you a little bit about the technology today so you understand the challenges that were involved in actually doing this. And I'm jumping to the end here, right, to give you sort of the punchline, and then we'll walk through some of the challenges. But ultimately, to engineer a yeast strain, right, so the yeast strains that we would use, let's say, to bake our bread or to brew our beer, to take a yeast strain like that and engineer it, so alter its metabolism so that it would grow on sugar and convert sugar, this very simple compound that you see here, to these complex medicinal compounds, basically required that we introduce 23 genes into that organism. These genes encoded basically 23 synthetic proteins that you wouldn't normally find within that organism. And those genes were taken from at least 6 different organisms that you could see here. So we took some genes from the opium poppy, but you can see that we also went into other plants as well, like the California poppy, and a medicinal herb plant, gold thread. We also went into mammalian organisms like the rat and bacterial organisms. And as I walk through some of the vignettes, you'll see why we did this, right? But again, it comes back to really leveraging the full biodiversity that's out there to identify the best activities that we want to bring together. So this is a pretty significant jump in scale in terms of complexity. You can think about recombinant insulin, right? as a drug, that required that we engineer a bacterial cell to make one protein, okay? And the protein was the product, the recombinant insulin. Here, you're now having to engineer a microbial cell to make 23 synthetic proteins. And you need those proteins to act together in an assembly line to basically take this sugar, break it down, and build it up to the molecule that you see here. So this is a much greater scale and complexity in terms of what we're talking about. So what are the challenges that come up when you do this type of research? The first challenge is that oftentimes the steps from sugar, let's say, to the medicinal opioid, the full set of steps, are not entirely known. So you have gaps in those pathways, which means you basically have to fill in those gaps to figure out how you can go stepwise from sugar to a medicinal opioid. The second is that you're taking enzymes from these very different hosts and putting them into a completely different host, the yeast. Those enzymes have evolved over many, many thousands of years, right, to act within a particular environment, let's say a plant cell. The plant cell presents a very different environment than a yeast cell. And so even if you get the protein made in yeast, oftentimes it's just not functional because the environment is so different. And this is sort of related just in terms of the complexity of the enzymes and the environments that they need to be present within the cell to have activity. So I'm gonna walk through just a couple stories that highlight the technologies that had to be developed to address these changes, okay? To address these challenges, I mean. And so I won't spend too much time on the slide, but this is basically zooming in on the sequence of steps that the yeast has to go through to take sugar and convert it to, let's say, a medicinal opioid like hydrocodone. For each one of the arrows that you see here is an enzyme catalyzed reaction, okay, or a protein that has to be made in that cell, okay? So you can see that there are a lot of arrows, right, as I showed you, and you can see that there are different color arrows. And so I'm going to refer to this as sort of a map when I talk about some of the challenges that we had to address. And when I first kind of zoom in on this part of the pathway, so because this was a really complex pathway, we basically broke it down into segments so that we can work on the different segments simultaneously, not just start at the beginning and go all the way through the end. And then as we got the pieces together, we could begin to assemble them. So this first challenge, or this first story, basically comes back to the challenge that I described in terms of not knowing the enzymes that are required to perform the specific steps, okay? And so if we zoom in on that part of the pathway, essentially what you see, and again, don't worry too much about the chemistry or the molecules, but the idea is that this is an amino acid, tyrosine, that yeast makes, right? So that's present in yeast. Everything else is not necessarily present in yeast, right? And so we need to start putting in synthetic enzymes. And so from the poppy and the different plants, these parts of the pathway were known. It was known what proteins did these reactions, but the proteins that catalyze the reactions from tyrosine to dopamine and this other molecule, 4-HPA, were not known. So we had to fill them in. Here we decided to take an approach that there's actually many, many organisms that can make dopamine. Humans make dopamine in our brain, right? So this is a very common molecule. So we don't have to be limited to the plant. Thank you. And so we can go into other organisms and basically begin to rebuild a synthetic pathway. And so that's essentially what we did. And so this is where it comes into us taking enzymes from mammals, and particularly we looked at human and rat enzymes to perform the reaction to make L-DOPA, which is again a molecule that our brains make. And then we chose a bacterial enzyme to convert L-DOPA to dopamine because it had really good efficiency within the yeast. And then it just so happened that there were a set of enzymes within yeast that could convert tyrosine to 4-HPA, so we just modified and overexpressed those. And then we essentially now were able to reconstruct the pathway that goes from tyrosine to reticulin. When we put the genes encoding those enzymes into our yeast, we grew our yeast on sugar, we could essentially show that yes, our yeast were able to convert glucose to this advanced intermediate reticulin, and this is just the analytical data that shows that our yeast are in fact making reticulin at reasonable levels. So that's just the first example of how, again, we can leverage the broader natural world, not just be limited to one organism, and begin to pull enzymes from very diverse organisms to rebuild pathways. The second story I want to go into is one that occurred later in the pathway as we begin to build and tailor the opioid scaffolds. And in particular, the challenge that came up here was one of context, right? So the different contexts that a plant presents versus what a yeast presents, and what you can actually see in terms of challenges with the pathway. So in particular, what I want to highlight, when I mean context, I want to highlight one particular example, which is plants are multicellular organisms, and they have cell types and they have tissue types, right? And what we know about the way that plants do their metabolism is they actually leverage different cell types to create different biochemical environments for certain reactions. So what you'll sometimes see is that when you have a long pathway, like what I showed you for the opioids, it'll break up the pathway into different cells within the plant. So let's say the first part of the pathway occurs in one cell type, they take that intermediate, they shuttle it over to the second cell type, and then it's acted on in the second cell type. Now, this is very different than when we just put everything in a microorganism like yeast, where all the enzymes are in one compartment, all the enzymes are seeing each other at the same time, and they're seeing all the intermediates in the pathway at the same time. And that can lead to a lot of different challenges. And one of the challenges that it can lead to is that you're no longer directing which enzymes are seeing which intermediates at a particular time. And so as you'll see, that can lead to a sort of lack of specificity in the pathway. So we decided to develop a tool set that would essentially mimic the kind of localization strategies that you can see in plants. And in particular, we know that cells are not just a bag of molecules. There's actually structure associated with them. And they have these intracellular compartments, as you see here. We can actually create genetic barcodes or genetic zip codes that we can attach onto our proteins. And those zip codes will basically direct the cell to direct the proteins to different compartments in the cell. And then in that way, you can begin to use those different compartments to achieve that type of biochemical specification and separation of different enzymes in the pathway. And we can show through microscopy that, in fact, we can localize our enzymes to particular compartments in the cell. So now let's go back and look at that part of the pathway. So here's the pathway zooming in, where we're decorating the opioid scaffold, starting with thebaine, going to morphine. When we put this into yeast, we saw something unexpected, which was we didn't just get this linear pathway that you see here, we got a branching effect, okay? And this would be undesirable if our goal was to make codeine or morphine, because essentially now about half of this product is being diverted to these undesired products and creating an impurity in the molecules that we're trying to make. And so again, this separate branching pathway wasn't observed in poppy. We thought about why this might be occurring in yeast, and we felt the culprit was the spontaneous reaction, basically not giving enough time for this compound, neopenone, to be converted to cotinone when everything was in the same cell. So we took an approach based upon delocalization, where we took this enzyme, core, and basically began to spatially separate it from the other enzymes in the cell. And I'm just showing you the data here. Again, the details aren't too important, but this is the base case where everything is in the same compartment in the cytoplasm. You see there's about a 50-50 split between neomorphine and morphine. These other cases are basically different cell lines where we localized this enzyme to different compartments in the cell, either the ER, the vacuole, plasma membrane, or mitochondria. And what you see is that that does shift the specificity. And there are several examples where we get pretty good specificity, up to about 90%, but still maintain the overall titers. So with that, we could take this type of strategy and then begin to ask whether we could go beyond just the compounds that poppy would make, right? So poppy makes morphine. We wanted to look at whether, by introducing other activities from other organisms, we could begin to make higher value compounds that you wouldn't find within the poppy. And in particular, we took a number of enzymes from a bacteria, Pseudomonas putida, which had been isolated growing on the waste from a poppy plant. So it had evolved enzymes that had activity on the opioids. We put these enzymes, more A and more B, into the plant, and essentially into our yeast strain. And what we could show then is that we could have yeast that would directly make hydrocodone, oxycodone, and hydromorphone, which are higher value semisynthetics that don't accumulate in the poppy plant. And again, we could grow our yeast strains, ferment them, and basically show that just by tailoring the combination of enzymes, tailoring our localization strategies, we could create different yeast strains that accumulated different profiles of the higher value semisynthetic opioids. Now, so that's sort of that story here. So basically what we had was this part of the pathway and this part of the pathway, and there were essentially two big remaining challenges that remained that we had to address. The first was that to go from S-reticulin to the morphinin structures, or to the opioid structures, there was an enzyme that basically did this reaction that had remained undiscovered. And for that one, we had to really go into the opium poppy because it was very specific to any organism that happened to make opiates, right? So we couldn't just look very much broader. And the second one was that the downstream enzyme, this enzyme, had extremely low activity in yeast. It had about 1% activity, which was not enough to sort of allow us then to assemble the full pathway and get pushed through a flux through the pathway to the desired product. So I want to just step through how we address these challenges really quickly. So the first one is discovering that missing link. So again, here we had a case, and we're zooming in on that part of the pathway, where the steps that allowed you to go from S-reticulin to R-reticulin were unknown. Based upon biochemistry that plant biologists had done over decades, it was hypothesized that this was a two-enzyme reaction with this intermediate that you see here. So we felt we were looking for two enzymes. The way that we approached this is that people had gone in and sequenced a number of medicinal plants, including the opium poppy and related poppy. So we developed a hypothesis about the type of protein that would be performing this reduction reaction here. And then we took a sequence that was homologous to that type of protein and basically searched those genome databases. But we specifically searched the genome databases for enzymes that were homologous to that reductase enzyme that we were proposing that were present within opium poppy, but not present within related poppy that didn't make morphine, right? So we're doing a cross search there. And it was very interesting to us because we got a number of hits, as you would see here, but in particular, there were a few hits from a couple poppies that made morphine that were particularly interesting because they were a fusion enzyme. What that means is that they were basically two proteins coupled together, okay? And one of those proteins was the reductase enzymes that we had proposed. And the second protein was a cytochrome P450, which could potentially be an enzyme that would catalyze this first reaction. And it was interesting because a fusion enzyme with a cytochrome P450, this class of enzyme, had never been discovered before in plants, so it was very unusual. But we thought, okay, this is pretty interesting. Let's take these enzyme sequences, right, from these databases that we uncovered. Let's resynthesize the DNA so that they can be made in yeast. And we put them into our yeast strain. And lo and behold, it was in fact the enzyme that was able to convert S-reticulin to R-reticulin. So we're able to discover a very novel new enzyme that performed this reaction that had eluded plant biochemists for decades. And in fact, instead of being two separate enzymes, it was one enzyme that was a fusion between two. And it was this very synthetic approach. So I find what's interesting about this approach and what it means in terms of how people can do this type of discovery is that we didn't actually have to have access to the poppy. We didn't have to have access to the physical material. Now that genome sequences are becoming open, databases that people can search, you now have access to that information, right, to begin to do these types of searches, develop hypotheses, and then use DNA synthesis just to reconstruct the sequence without ever having to access the actual organism that you're taking those sequences from. Now, the final thing then was this final protein. So we discovered this enzyme, and we still had this enzyme, which, as I said, had about 1% activity, and we had to fix it. So we had to do some detective work, and basically what was challenging is that this is an enzyme that does have to be localized to an endomembrane within yeast. When we looked at a protein gel to see if the protein was being made in yeast, it was. But when we did a more careful analysis, what we saw is when we compared the banding to what the protein looked like in plants, you could see that there were sort of several bands that were showing up, which wasn't the case to how it was being expressed in plants. And this was indicative that the protein was actually being made, but it was being misglycosylated, or basically mismodified. So when it gets mismodified, right, what happens is that it gets incorrectly inserted into the membrane. So instead of facing one way, it faces the other, and that very substantially impacts the activity of this protein. So we developed a strategy of engineering the protein where we basically developed a chimeric protein between taking the beginning part of a related protein that we knew was processed correctly within yeast and fusing it to the catalytic part of this protein. And we could show then, these are just variants of the fusion proteins that we made, but basically we can show that creating that fusion did in fact increase the activity of the protein, and we could find particular fusion strategies that would increase the activity by about 10-fold, which was sufficient for us to assemble the entire pathway. So at the end of this then, again, what we had was an organism that was making 23 synthetic proteins and was able to grow and ferment on sugar, and within a matter of 3 to 4 days, make 2 different opioid compounds, hydrocodone directly, and then thebaine. So I talked to you a little bit, or I mentioned earlier this idea of having a platform, right? That once we do this for one class of compounds, we can take the technology and very quickly apply it to a different class of compounds. We've been able to show that. And so specifically, again, what I mean is that in this case, we had a platform strain that made this S-reticulin compound. And to access now, let's say, compounds that are of use to treat cancer or potentially neurodegenerative diseases, we can take that optimized platform strain and just modify the downstream enzymes using the tools and strategies that I just described to you proactively. And then what we're able to show is that within less than a year of a single postdoc's time from developing the opioid strains, we could now have strains that would make noscapine and related derivatives to noscapine that are of interest for these other types of activities that I described. So I want to end just by discussing a little bit, highlighting the pace of change in the field, because I think now what we're seeing is that the field is moving very, very quickly. We started this work in general in my laboratory about 10 years ago. And at that time, the tools and techniques that I described to you were really not in place. And so it took us a very long time to get the sort of first working strain for opioid production. But it was not a linear progression across those 10 years. It was really sort of exponential, where I would say within the past 2 or 3 years, as the tools, some of which I've highlighted to you today, have come together, we've been able to make very, very rapid progress in putting all the pieces together. And then, as I mentioned, what you saw is that once we were able to do that first demonstration, we could very rapidly and with minimal effort expand it to a second class of compounds with very diverse therapeutic profile. And in fact, we've been able to expand it even further to make an even larger number of different plant alkaloids by leveraging both the platform technology and the tools that I described. Now, another question that comes up is sort of when will this technology actually be entering the market? So what we described last year in the paper that we reported, sort of the first description of yeast strains making opioids, was really a proof of principle, meaning that those yeast strains were making opioids at concentrations that were too low to be useful commercially, right? You wouldn't take them into commercial scale-up. We estimated at the time that we would have to increase titers and productivity by about 100,000-fold. So since that time in September, we've now increased it by about 1,000-fold, and we have 1,000-fold left to go. And so we predict that by the end of 2017, those strains will actually be ready to enter commercial scale-up, meaning that you can actually take them into large-scale manufacturing facilities and begin to produce these molecules. And we further estimate that they will reduce the cost of production by over 10-fold. Okay, so again, one of the things I want to leave you with here today is the idea that what I've described to you is not just one example of how we've been able to engineer a strain to make one particular class of compounds and address some of the challenges that are inherent in the ways that we source and make our pain medicines, but that in fact, by addressing the challenges, by developing the technology and addressing the challenges for one of the most kind of complex test cases out there, we now have the tools, knowledge base, and technology in hand to begin to apply these to a wide diversity of different types of compounds that will address many, many different types of diseases. And that's the real impact of the technology that you see here today. So with that, I will end and thank the researchers who have done the work. This has been a long effort in my laboratory, and so I'll just highlight some of the people in particular. Kate Thody, who did a lot of the work on the downstream engineering and the delocalization strategies to get to natural and semi-synthetic opiates. Isis Trenchard and Mike Siddiqui, who did a lot of the engineering of the first upstream part of the pathway to rebuild the pathway to get to S-reticulin. And then Stephanie Galini, who did a lot of the work on the discovery of that new enzyme that was the gatekeeper to the opioids, and also optimizing the activity of that cytochrome P450. And I will end here and take any questions. Thank you.
And if you have questions, please go to the microphones so that we can capture them.
Hi, I want to start off by saying that's some of the most elegant research I have ever seen.
Thank you.
I wondered, again, if you could explain why, or the benefits of making two compounds at the same time, or was it just you wanted to challenge yourself? Why make, because then you have to separate them.
So yeah, let me be a little bit clear. So yes, the question is, what are the benefits of making two compounds at the same time? So let me step back and say the 2 compounds that we made in that paper were from 2 separate strains. So one strain was making thebaine, which is a starting material for a lot of the semi-synthetics, so there was value in making that. The second strain was making hydrocodone. Okay, so it was 2 separate strains, not both in the same strain. However, what we now, with our current strains, which are non-optimized, they do accumulate not all the intermediates along the pathway, but they'll accumulate several intermediates along the pathway. And you're right, ideally you would like your strains to primarily make one, because that makes the downstream separation process much easier, right? So as we do the work of sort of improving these strains and optimizing them to get to the productivities and titers that we would like, we're also trying to make sure that they're primarily only accumulating the target opioid. And the idea is that we would have different production strains for different target opioids to go after.
Got it. Now also, since you had seemingly 20 or 30 or 40 reactions, I mean, it was incredibly elegant that you did it in different portions of the cell membrane, different portions of the cell. But to what extent did reaction kinetics play a role in all of this, especially since you did have multiple reactions going on at the same time? How were you able to control the reaction kinetics?
Well, you do have multiple. So the question is, how do reaction kinetics play into this? And the answer is they play quite a significant role, right? All the reactions are going on at the same time, right? And so you can imagine the yeast is growing, right? And so you first are building biomass, and then the yeast basically enters kind of a stationary phase where it's no longer accumulating biomass, and that's where you hope it's really cranking on the production of these compounds. And so how does kinetics play a role? It plays a substantial role because these different enzymes will have different kinetic properties. Some will be slower than others, right? And the ones that are slower than others can create challenges. Where they create challenges primarily is in the fact that the yeast, so one thing I didn't mention is that these compounds primarily accumulate outside of the yeast, okay? And that's because the yeast is pumping them out. It doesn't like to keep them inside. And yeast have transporters that specifically act to kind of pump out compounds like this. On one hand, that's great because it means that the separation process is pretty easy. We don't have to break open the cells. We can just settle them and then purify things from the media. On the other hand, if you have slow enzymatic steps, it becomes a challenge because that intermediate will accumulate in the cell and get pumped out, right? And then once it's pumped out, it can't be acted on by downstream enzymes. So for the enzymes that are slow kinetically, we basically do protein engineering to increase their kinetics and increase their reaction efficiencies. And so far, we've been finding that that works. But it is something that you have to address.
Cool. I've also seen you filed a number of patents. Has the patent process been as challenging as the research?
That's a good question. So yes, we do have a number of patents. And I guess I would say this. I don't think it's been as challenging as the research, but I also think it's very dependent on, I found it to be very dependent on the lawyers that you're working with. For example, these are all filed through universities that I've been at. I was first at Caltech, so we had a patent or 2 filed there, and then at Stanford, and there are a number of patents filed there. And then we've started a company about a year ago to commercialize a lot of the technology that I showed you here today. And so the company was able to license the patents, and it also got its own attorneys to handle the patents. And I found that once we actually had our own attorneys to handle the patents, and we're paying them a good deal of money, it was incredible how much faster the process went, and also how much strategy was put in play in terms of thinking through the claims and how to think through the entire portfolio, et cetera. So I would say within the past year, it has felt a lot, not easier, but just, kind of easier.
So it sounds like you used kind of the same strategy. You had individuals, you focused on this particular process instead of having a whole cell do it or a whole team of different attorneys from outside. You focused on one area. So it seems like you had a parallel between your patent process and your science.
Yeah, there is a parallel. There is a little bit of a parallel, right? Especially when you're at a university, the university is filing the IP and the university owns the IP and they're going to use their particular attorneys, right, that they're going to use. And then once a company licenses it, right, then they're more than happy usually to allow the company attorneys to kind of take over, assuming they're doing the right thing, and put more effort and time and thought into it. So that's been my experience from both universities that I've been at.
Thank you very much, Professor Smolke.
Yeah. Oh, one question. Yes?
Yeah, thank you for the talk. I was wondering if you have run into any government regulations, since to me, you were talking about, these can be made into, I forgot, like heroin or something, so bad drugs in the beginning, and I could imagine someone working in a company making these opioids to like steal a vial of these tiny little yeast, and then just go home and put it into a big vat and make lots and lots of drugs very easily and much quicker, like you said. So I'm wondering if like the government has...
So this is a great question, and actually I'm just going to go, because this question comes up a lot. So first thing is, there is regulation associated with these compounds, specifically at the level of DEA, right, in the US. Every country has its own regulatory body. So for us to be able to make these strains and work with these compounds in my laboratory or in a company, I have to be legally, I have to be registered with the DEA. So they have to know what we're working with, and what they really care about is monitoring and tracking and diversion, right? So you have to have a lot of processes in place to avoid that, right? Both in terms of the physical barriers and then in terms of just the processes and who's able to handle them, and background checks and everything else. So that definitely has to happen. Now, there's been, and that's true, for anything. Now, and if anybody requests these strains from us, we have to, from my academic lab, right, we have to basically confirm that they have the appropriate approvals from the relevant regulating body to be working and doing research with these compounds. The second part of your question was, this idea that, okay, these are yeast. We know how to grow yeast. People have homebrew kits, What's stopping someone from like stealing a little bit of a strain, taking it to their house and just, growing tons and tons of yeast in a homebrew kit or in their bathtub and making lots of compound? And so one of the things that's often overlooked is the role of process versus the role of just biology. And what I mean by that is that when we grow these in the laboratory, or when a company grows them, right, for production, you're growing them under very, very tightly controlled conditions, right? So the way that we ferment compounds for production like this looks very different than the way that we homebrew. Everything is controlled, from the way that you feed glucose, from the way that you feed oxygen, the pH, everything. And that control plays a really large part in how they make and how much of these compounds the yeast can make. So we actually did an experiment to demonstrate this, where we took the yeast strains that I described, we grew them as we would in the laboratory, which is what you see on the left, these small little fermenters where everything's being controlled, et cetera. And then we did a homebrew experiment in the laboratory, where we took the yeast and we just threw them into kind of the starting material that you would use for a homebrew, right, where everything's there at once, and you just kind of let them grow. And what we showed is that basically you observe no production of the opiates in the homebrew setting, okay? And the farthest compound along the pathway that we were able to see was that reticulin compound, and it was at much lower concentrations, okay? So it is not to say that once we increase the productivity 100,000-fold, you're not going to get, some amount of these compounds being produced from yeast if you grow them that way. But it is to say that it's going to be substantially less than what you would get to make it kind of commercially interesting, from the industrial perspective. And then the question is really, there are so many other ways right now to get these compounds, whether it's from poppy seeds, and brewing tea, to just even, abuse of legal medicinal opioids that are frankly, at least right now, a whole lot easier and cheaper than going through this process of, whether somebody would actually be motivated to do that at that level, right? So, and again, it's not to say that absolutely no, but it is to say I think there's a lot of questions in terms of how realistic that particular scenario is. But it's something that we definitely want to be looking at and thinking about as the technology develops.
Okay, one final question.
Okay.
Actually, two quick questions.
One final question.
It sounds like you engineered the first 1,000% increase, are you going to consider using, seems like resistance, there's a lot of genetic information out there that you could use through plasmids and artificial selection. Do you have a means to selectively survive the maximum expressors of your morphine or whatever and breed and use nature, mix it with a bunch of highly resistant bacteria that have these abilities to express large amounts of compounds?
Do you mean in terms of resistance in terms of what? Resistance in terms
like
of?
antibiotic resistance,
Oh.
things like that, right?
Yeah, so when we engineer our yeast strains, we typically actually will not express things off of plasmids. We put things into the chromosome, which is really easy to do with yeast.
Right.
And when we put things into the chromosome, it becomes fairly stable, meaning that we don't have to actually put resistance pressure on the yeast to grow, so they can just grow in regular media, and they're fine. And that's desirable because it basically, and then we can remove the antibiotic resistance cassettes, et cetera, so they're just sort of wild-type yeast strains, right? Or they're yeast strains that don't have those types of resistance pressures. Number one, I think that makes the fermentation a little bit cheaper because you don't have to add antibiotics and other things to the fermentation or worry about those types of waste streams. It also makes the cells more genetically stable.
I guess the second question is, if this seems like $1 billion breakthrough, that list of graduates and postdocs that you thanked, is any of the money going to flow through to them, or did they just get a plaque?
So that's a great question. And I think it does in several different ways, right? The first way is that they are inventors on the patents, right? So, and of course the universities own the patents, but as inventors on the patents, they get a certain share of, the revenue and royalty that comes in from the patents through the university, right? Typically, most universities, I think it's like a third split, right? Where a third goes to maybe the university TLO, a third goes to the department, and then a third goes to the inventors. And, so whatever licensing agreements are in place in terms of those percentages, right, with the royalties, et cetera, that amount then would go to them, whether or not they're involved with the technology beyond this or not. So that's the first thing. And then the second thing is that, we did start a company on this, as I mentioned, and there are 4 co-founders of the company, myself, and then 3 others. 3 others are 3 of the people that I named who had led this work, right? And so they're very active in the company, taking it forward, helping to build the culture, the climate, and they are going to progress into, leadership roles within the company and hopefully be very successful and help make the company successful. And most all of the people who worked on it had the opportunity to kind of do that, where a significant percentage decided that that was what they wanted to do. Okay. Thanks.